Hello, I'm Sean Kim from Seoul National University, and I'm presenting a paper entitled CReSE, Benchmark Data and Automatic Evaluation Framework for Recommending Eligibility Criteria from Clinical Trials Informations and EACL 2024 Findings. In this paper, we define the problem of recommending eligibility criteria, we call EC in these presentations, in clinical trials, based on free text descriptions such as trial titles and summary informations as an NLP test, and propose related benchmark data. In order to evaluate the performance of EC recommendation model from clinical perspective, we develop an embedding system named CReSE that reflects the clinical relevance between EC and propose an automatic evaluation framework. First, talk about our study motivations and contributions. As you might know, clinical trials are gold standard research methodology for evaluating the efficacy and safety of medicines and exploring causal relationships between medical interventions and treatment effects. So one of the key design factors of clinical trials is EC eligibility criteria, which specifies the conditions of patients who will be included in a given trials. So ECs are primarily divided into inclusion criteria, which specify which patients will be included in the study, and exclusion criteria, which specify which patients will be excluded. Also, EC cover a variety of clinical and social factors such as age, gender, underlying medical conditions, and medication history. ECs are mainly designed for three purposes. So first and most importantly, they are designed to minimize risk and protect patients in clinical trials. Also, by ensuring that patients with similar clinical situations are included in the trials, so trial designers, mostly pharmaceutical companies, aim to increase the statistical power of clinical trials. However, as effort to increase the homogeneity of patients in clinical trials have long been ongoing, there has been a growing concern that the patients in clinical trials are not represented enough. As noted in the following FDA guidance, EC that are not based on the adequate clinical justifications are frequently used in current clinical trials because they are customary and have been used in similar trials in the past. These restrictive ECs can lead to clinical findings not being adequately generalizable to real-world care settings, and also have health equity implications in that they prevent clinical evidence from being collected in minor subgroups such as children, older adults, or patients with HIV infections. In this context, we define the task of recommending eligibility criteria as an NLP test and build a benchmark data set for the test. By doing so, we expect that we possibly provide EC template that better meet the purpose and settings of clinical trials under design from basic trial information like trial titles. Also, we develop an embedding system that reflects the clinical semantic similarity between different ECs and propose an automatic evaluation framework for evaluating EC recommendation models from clinical perspective. By utilizing the embedding system we developed it. We named the text embedding system as CReSE. So before we dive into discussing detailed methods and results of our study, let's briefly review the previous NLP studies related to clinical trial design. To date, NLP studies attempting to facilitate clinical trial design have been conducted under two main streams. One direction is the group that attempts to obtain appropriate template design trials by searching for trials similar to one they wish to design, and the other is a group that attempts to generate ECs that are ready to use in a trial setup. So especially in recent years, with the rapid development of LLM, attempts has been made to generate specific EC conditions that are appropriate for clinical trial setup by utilizing language models. However, to date, EC generation studies still rely on manual instructions from clinical expertise to generate specific EC. Another limitation is that dataset and models have been developed primarily for a limited scope of EC conditions such as patient gender or age, BMI, QT interval like that. Moreover, previous EC generation studies have evaluated model performance by comparing generated EC and ground truth ECs using traditional summarization evaluation metrics, or by converting ECs into the query use pre-developed EC parser models. However, this is not a good way to evaluate the performance of the EC generations and recommendation models, because in clinical situations, sentences with very different words often refer to the highly similar clinical situations, as shown in the following examples. Furthermore, although the performance of EC parsers have improved a lot, they still have a limited parsing performance on complex ECs, and there is still no consensus on a grammar for parsing ECs. In this context, we focus on proposing an EC recommendation task and developing an automatic evaluation framework for the evaluating the EC recommendation model from the clinical perspective. So now let's talk about our study method and results. We collect the clinical trial information from clinicaltrial.gov to develop benchmark data set for the EC recommendation task. Clinicaltrial.gov is the largest and most comprehensive clinical trials registry operated by the U.S. National Library of Medicine, NLM, and currently has collected information on more than 440,000 clinical trials in more than 200 countries. So you can think of it as a registry where almost every normally conducted clinical trials is registered. Based on our expertise in clinical trials, we select clinical trials and EC included in the benchmark data set to ensure the EC recommendations are made in the consistent context, resulting in a more appropriate clinical advice. First of all, because in clinicaltrial.gov, information about a huge variety of clinical trials has been uploaded, so we constructed our data set using only interventional trials for drug interventions. In addition, we used the trials where key essential information was reported and the number of ECs was not too small to ensure that the quality of reporting of the trial information. We also use only ECs of appropriate length based on the heuristics, as ECs that are too short or too long may have unclear clinical meanings or include multiple conditions at once. So, because we aim to recommend ECs when the trial setup was given by the trial title and summary text, we needed to exclude ECs that are used in almost every trial, regardless of the trial setup. Therefore, based on the existing FDA guidance, we define ECs that are frequently used regardless of the setup of clinical trials as common ECs and develop a classification model for the common ECs. For example, ECs such as the include only patients over 18 years old or exclude pregnant and breastfeeding women are examples of common ECs. So we obtained the relevant training data set in form of simple binary classifications and perform the BIRD fine-tuning to obtain the high classification performance of affluence scores of over 97 in BioLink BIRD. So using this common EC classifier, we were able to formulate a clinically meaningful EC recommendation task by excluding common ECs from the entire data set. And to provide the quality of negative EC title pairs in the benchmark data set, we took the following steps to exclude these common ECs, as a large number of ECs are still using in a different trials. In the end, we developed benchmark data set for the development of high quality EC recommendation models with nearly 1.7 million non-common ECs from a total of 270 case trials and equal number of the negative EC title pairs. As described earlier, in this study, we also developed an embedding system named CReSE that reflects the clinical relevance between ECs. So for developing this model, we rephrase ECs using the latest LLM and perform the contrastive learning use the original rephrased EC pairs. We also developed 4 different rephrasing prompts so that various clinical knowledge can be utilized in the EC rephrasing processes. So we evaluate the CReSE model by generating clinical relevance data by using the LLM or human expert. Yeah. So we get the clinical relevance data of the arbitrary EC pairs and measure the correlations between the similarity, cosine similarity in the embedding systems of 2 ECs and their clinical relevance. So as a result, CReSE model developed in this study outperformed various state-of-the-art biomedical language models in EC clusterings and reflects semantic similarity in the biomedical domain. Furthermore, we confirmed through evaluation study that it is important to utilize the variety of rephrasing prompts when training the CReSE model rather than having a larger number of original rephrased EC datasets. Also, we found a weak inverse correlations between the EC clustering performances of CReSE models and the training losses on the contrastive learning. We believe that these correlations suggest that EC rephrasing itself is an effective instruction to extract knowledge about clinical relevance from LLMs, but at the same time, the objective between the contrastive learning and EC clustering are different, so the prompt engineering to match these 2 tasks may be important. So finalized EC recommendation models show up to 90% accuracy in binary classification task formulations, which greatly exceeds the performance of ChatGPT and GPT-4. Even when all ECs are clustered into the 100 clusters, the top 1 precision was close to 50%, showing a high performance. In qualitative review, we found that the ECs were appropriately recommended for the investigational drugs or disease conditions described in the trial titles. However, in many cases, the purpose of studies or specific patient conditions is not fully expressed in the study titles, so it might be necessary to provide additional information such as brief summaries or design factors for the achieving higher recommendation performance. To summarize, in this study, we defined the task of recommending ECs from basic trial information as an NLP task and published high-quality benchmark dataset. Also, we developed a CReSE model that reflects the clinical relevance of ECs and proposed an automatic evaluation frameworks based on the CReSE models. The code and data used in this study can be found on the GitHub and Hugging Face, and we hope that this study will contribute to provide a better EC templates for the clinical trial designers so that the clinical trials can be conducted in a more representative way. So thank you for listening to our presentation. Thank you.